Hi class, this is your instructor, Skylar Huff, and we're now in Chapter 28, Protists. For more than half of the history of life on Earth, all life was microscopic. For over 2 billion years, the largest organisms were unicellular prokaryotes. These prokaryotes lacked internal membranes except for folds in surface membranes of photosynthetic bacteria. The first evidence of a different type of organism is found in tiny fossils in rock 1.5 billion years old. These fossil cells are up to 10 times larger than bacteria and contain internal membranes and what appear to be small membrane-bounded structures. The complexity and diversity of form among these single cells is astonishing. The step from relatively simple to quite complex cells marks one of the most important events in the evolution of life, the appearance of a new kind of organism, the eukaryote. Protists are a paraphyletic group of eukaryotes that are classified as animals, plants, or fungi. Although the group may seem a little strange at first, its members, like the unicellular green alga, Macrosterias, pictured here, play an important role in human health and welfare, ecology, and nutrient cycling in the environment. Protists were the first eukaryotes. Eukaryotic cells are distinguished from prokaryotes by the presence of a more complex cytoskeleton and compartmentalization. The exact sequence of events that led to large, complex eukaryotic cells is unknown, but several key events are agreed upon. Loss of a rigid cell wall allowed membranes to fold inward, increasing surface area. Membrane flexibility also made it possible for one cell to engulf another. In rocks about 1.5 billion years ago, scientists have found well-preserved microfossils that are noticeably different in appearance from earlier, simpler cells. An example of fossil algae is shown in this figure before you. These cells are much larger than those of prokaryotes and have internal membranes and thicker walls. Early fossils mark a major event in the evolution of life. A new type of organism had appeared. These new cells are called eukaryotes, from the Greek words meaning true nucleus, because they possess an internal structure called a nucleus. All organisms are either prokaryotes, which lack a nucleus, or eukaryotes, which possess a nucleus. During discussions of the origin of eukaryotic internal structure, keep in mind that as discussed in Chapter 24, horizontal gene transfer occurred frequently while eukaryotic cells were evolving. Eukaryotic cells evolved not only through horizontal gene transfer, but also through infolding of membranes and by engulfing other cells. Today's eukaryotic cell is the result of cutting and passing of DNA sequences and acquisition of organelles from different species. Many prokaryotes have infoldings of their outer membranes extending into the cytoplasm that serve as passageways to the surface. The network of internal membranes in eukaryotes is called the endoplasmic reticulum and the nuclear envelope, an extension of the ER network that isolates and protects the nucleus. It is thought to have evolved from such enfoldings, as you see here in Figure 28.2. Bacteria that live within other cells and provide some benefit for their hosts are called endosymbiotic bacteria. Endosymbiosis means living together in close association. Their widespread presence in nature led biologist Lynn Margulis in the early 1970s to champion the theory of endosymbiosis, which was first proposed by Konstantin Merkowski in 1905. The endosymbiotic theory supports the concept that a critical stage in the evolution of eukaryotic cells involved endosymbiotic relationships with prokaryotic organisms. According to this theory, energy-producing bacteria may have come to reside within larger bacteria or archaebacteria, eventually evolving into what we now know as mitochondria. and you can see that here in figure 28.3. One scenario posits that the original host was an anaerobe and the hydrogen-dependent metabolic pathways, and that the symbiont produced hydrogen that could be used by the host. Later, the host adapted to an oxygen-rich environment using the symbiont's respiratory pathways. Photosynthetic bacteria were likely engulfed by other larger bacteria, leading to the evolution of chloroplasts, which are the photosynthetic organelles of plants and algae, shown to you in the same figure, 28.3 here. The history of chloroplast evolution illustrates that care must be taken in phylogenetic studies. All chloroplasts are likely derived from a single line of cyanobacteria, but the organisms that host these chloroplasts are not monophyletic. This apparent paradox is resolved by considering the possibility of secondary and even tertiary endosymbiosis. Figure 25.10 explains how red algae both obtained their chloroplasts by engulfing photosynthetic cyanobacteria. The brown algae most likely obtained their chloroplasts by engulfing one or more red algae, a process called secondary endosymbiosis, shown to you now in figure 28.4. A phylogenetic tree based only on chloroplast gene sequences from brown, red, and green algae reveals a close evolutionary relationship. This tree is misleading, however, because it is not possible to tell just from these data how much the algal lines had diverged at the time they engulfed the same line of cyanobacteria. Nuclear gene sequences, as well as morphological and chemical traits, are more helpful than chloroplast gene sequences in sorting out algal. That we now witness so many symbiotic relationships leads general support to the endosymbiotic theory. Even stronger support comes from the observation that present-day organelles, such as the mitochondria and chloroplasts, contain their own circular DNA, which is remarkably similar to the DNA of bacteria in size and organization. During the 1.5 billion years in which mitochondria have existed as endosymbionts within eukaryotic cells, most but not all of their genes have been transferred to the chromosomes of host cells. Also, the mRNAs produced from the genes of the mitochondrial genome are translated using ribosomes that are very much like ribosomes in size and structure. Many antibiotics that inhibit protein synthesis in bacteria also inhibit protein synthesis in mitochondria and chloroplasts, but not in the cytoplasm. Further supporting the theory of endosymbiosis, chloroplasts and mitochondria replicate by binary fission just as bacteria do. A prokaryote carries its genes in a circular DNA molecule. In contrast, a eukaryote carries its genes on multiple chromosomes, which are usually present in pairs. During the evolution of eukaryotes, a systematic process needed to be developed to divide the chromosomes and other cell contents during cell division. The separation of chromosomes is called mitosis, whereas the division of the cytoplasm is called cytokinesis. In fungi and in some groups of protists, the nuclear membrane does not dissolve during mitosis, as it does in plants and animals, and most other protists. Consequently, mitosis is confined to the nucleus. When mitosis is complete in these organisms, the nucleus divides into two daughter nuclei, and only then does the rest of the cell divide. In all other eukaryotes, the nuclear membrane breaks down prior to mitosis, so the separation of chromosomes occurs in the cytoplasm rather than in the nucleus. We do not know whether mitosis without nuclear membrane dissolution represents an intermediate step on the evolutionary journey, or simply a different way of solving the same problem. We cannot see the interiors of dividing cells well enough in fossils to trace the history of mitosis. Unlike other groups of organisms, protists have no unifying features, but rather are characterized by diversity in structure, genetics, reproductive strategies, and metabolism. Unlike the way many organisms are classified, a eukaryote is classified as a protist by exclusion. That is, a eukaryote is considered a protist if it lacks the characteristics required to place it with the other fungi, plants, or animals. Many protists are unicellular, but numerous colonial and multicellular groups also exist. Most are microscopic, but some are as large as trees. The origin of eukaryotes, which began with ancestral protists, is among the most significant events in the evolution of life. Eukaryotes diverged rapidly in a world that was shifting from anaerobic to aerobic conditions. We may never be able to completely sort out the relationships among different lineages during this major evolutionary transition. Applications of a variety of molecular methods are providing insights into the evolutionary relationships among protists. Molecular systematics is helping to sort out the protists, which are now classified into 5 supergroups: the Exuvata, the SAR comprising of Straminophila, the Aviolate, and the Rhizaria, the Archaeplastida, the Amoebozoa, and the Opisthocanta. In efforts to resolve the controversies surrounding protist phylogeny, DNA sequence data are being used to test different hypotheses regarding evolutionary relationships within and between the groups. The phylogenetic tree shown in Figure 28.5 represents one of several organizations regarding the relationships between protist lineages, but is but one hypothesis. In this chapter, we explore the diverse world of protists based on our current understanding of phylogeny. Understanding the evolution of protists is key to understanding the origins of plants, fungi, and animals. The supergroups encompass all eukaryotic diversity, and many protists are closely related to plants, animals, or fungi. The remaining chapters in this diversity unit, taught in Principles of Biology 2, focus on the monophyletic fungi, plants, and animals that trace back to common ancestors shared with different protist lineages. Protists possess a varied array of cell surfaces. And let me pause here for Figure 28.5. So in this figure, you're shown the eubacteria, root of the tree. You're shown thereafter archaea. And then from there, we have these 5 supergroups, which include all eukaryotic life, and of course the protists. So if you look at Archaeplastida, you see the land plants, among other photosynthetic. Thereafter, you see Opisthocanta, and you have their fungi and animals. Protists possess a varied array of cell surfaces. Some protists, such as amoebas, are surrounded by only their plasma membrane. All other protists have a plasma membrane with an extracellular matrix deposited on the outside of the membrane. Some extracellular matrices form strong cell walls. For instance, diatoms and foraminifera secrete glassy shells of silica. Protists have several means of locomotion. Protists move by diverse mechanisms. Protist movement is chiefly either flagellar rotation or pseudopods. Many protists wave one or more flagella to propel themselves through the water, and others use banks of short flagella-like structures called cilia to create water currents for their feeding or propulsion. Pseudopods are the chief means of locomotion among amoebas. Other protists have long, thin pseudopods that can be extended or retracted. Because the tips can adhere to surfaces, the cell can move by a rolling motion, shortening the pseudopods in front and extending those in rear. Other protists have numerous cilia that act as tiny oars to move the protist through fluid. Protists can be heterotrophic or autotrophic. The heterotrophs obtain energy from organic molecules synthesized by other organisms. Some autotrophic protists are photosynthetic, whereas others are chemoautotrophic. Some heterotrophic protists are phagotrophs. These are organisms that ingest particles of food into vesicles called food vacuoles or phagosomes. Lysosomes fuse with food vacuoles, introducing enzymes that digest the food particles within. Digested molecules are absorbed across the vacuolar membrane. Another example of the protist's tremendous nutritional flexibility is seen in the mixotrophs, protists that are both phototrophic and heterotrophic. Protists typically reproduce asexually. In addition, some undergo sexual reproduction regularly, whereas others undergo sexual reproduction at times of stress, including during food shortages. Asexual reproduction involves mitosis, but the process often differs from the mitosis in multicellular animals. For example, the nuclear membrane often persists through mitosis, with the mitotic spindle forming within the nucleus. In some species, a cell simply splits into nearly equal halves after mitosis. Sometimes the daughter cell is considerably smaller than its parent cell and then grows to adult size, a type of cell division called budding. In schizogony, common among some protists, cell division is preceded by several nuclear divisions. This allows cytokinesis to produce several individuals almost simultaneously. Most eukaryotic cells also possess the ability to reproduce sexually, something prokaryotes cannot do. Meiosis is a major evolutionary innovation that arose in ancestral protists and allows for the production of haploid cells from diploid cells and the generation of genetic diversity. Sexual reproduction is the process of producing offspring by fertilization, the union of two haploid cells which were generated by meiosis. The great advantage of sexual reproduction is that it allows for frequent genetic recombination, which contributes to the variation required for evolution by natural selection. The evolution of meiosis and sexual reproduction contributed to the tremendous explosion of diversity among the eukaryotes. Diversity in eukaryotes was also promoted by the development of multicellularity. Some single-celled eukaryotic cells began living in association with others in colonies. Eventually, individual members of the colony began to assume different duties, and the colony began to take on the characteristics of a single individual. Multicellularity has arisen many times among the eukaryotes. Practically every organism big enough to be seen with the unaided eye, including all animals and plants, is multicellular. The great advantage of multicellularity is it fosters specialization. Some cells devote all their gene energies to one function in tissue, other cells to other functions. Few innovations have had as great an influence on the history of life as the specialization made possible by multicellularity. So let's get to our first group, the excavata. So to be brief, diplomonids lack functional mitochondria. Diplomonids are unicellular. They move with flagella and have two haploid nuclei. Parabasalids have undulating membranes. They use flagella and undulating membranes for locomotion. So there is a particular type of parabasalids, and the type I'm thinking about would be the trichomoniasis, and this parasite species I think of here causes an STD. And then finally, the euglenozoans. So the euglenozoans, they change shape while swimming. They're free-living euglenids, and they're exemplified by euglena, and they can produce chloroplasts to carry out photosynthesis in light, and they contain a pelicil and move via anterior flagella. And then they also have the chenoplastids and are parasitic and are distinctive in having a single unique mitochondrion with two types of circular DNA. So as mentioned once again, some have chloroplasts that may become heterotrophic in the dark. They may become heterotrophic in the dark. And that is the euglenids by way of euglenozoa. Just one more time. A third of the euglenids have chloroplasts and are autotrophic, and the others lack chloroplasts and ingest their food and are heterotrophic. This is it. And the second major group are the parasitic kinetoplastids. And those are the ones that are distinctive and have that single unique mitochondria with two types of circular DNA. And included in this groups are the trypanosomes. So the trypanosomes cause human diseases. So one type would be the African sleeping sickness from the tsetse fly, and then the other type would be leishmaniasis, the sandfly, and then Chagas disease, which you can get from that skin contact with urine or blood of an infected wild animal. So you have that trypanosome shown here with this blood, the SAR supergroup. So here we have the straminophils, the alveolates, and then the rosaria. This is where we are, SAR. So here we have the brown algae, the diatoms, and the oomycetes. Brown algae are typically large, and I say large, and as I mentioned, large seaweeds that have haplodiplotontic life cycle, producing a gametophyte and sporophyte stage. So the gametophyte is the haploid multicellular state, and the sporophyte is that multicellular diploid state. These are not plants. They're brown algae. The diatoms. They are unicellular organisms with double shells. As you see here, they are beautiful. So diatoms have silica in their cell walls, and each diatom produces two overlapping glassy shells. And they fit like a box and lid. And lastly, we're at the oomycetes, or water molds. So they have some pathogenic members. So the oomycetes are parasitic and are unique in the production of asexual spores, or zoospores, that bear two unequal flagella. So the water molds, they're wholly different, as they were once considered fungi. So they undergo sexual reproduction, and they're found in water and on land. So one thing to mention about the water molds is the Phytophthora infestans, which causes late blight of potatoes, was one of the multiple factors, including weather and the oppressive social conditions, that contributed to the Irish potato famine of 1845 to 1849. And during the famine, over 40,000 people died, starving due to diseases by way of that complication. And about 2 million Irish immigrated to the United States and elsewhere. Next up, we get to alveolata, also part of SAR. So here we'll begin with the dinoflagellates. From there we'll go to the apicomplexans and then end up with the ciliates. The dinoflagellates are photosynthesizers with distinctive features. The dinoflagellates have pairs of flagella arranged so that they swim with a spinning motion. So blooms of the dinoflagellates are what causes red tides, which I think you all have heard of. So that red tide is that explosion of these as they're growing. So what happens is, well, the fish, the birds, and marine mammals, they typically die from those toxins due to those red tides, as one is shown to you here. The apicomplexans, they form spores, so that they have that apical complex, and it's a unique arrangement of organelles at one end of the cell. So they have a unique arrangement here, and it's called the apical complex, and this is what is used to invade the host. So the best known apicomplexan is the malarial parasite Plasmodium. Malaria, which was discussed in chapter 24, is a disease with 218 million cases and 405,000 deaths in 2018 were caused by the Plasmodium parasite. So the parasite is transmitted via bites of infected Anopheles mosquitoes. So yes, it's a vector-borne disease. malaria is. So Plasmodium has a complex life cycle involving sexual and asexual phases, alternating between different hosts. In this case, the mosquitoes, the Anopheles gambiae, and humans. And you're seeing here how it works. So while feeding, the mosquito injects the Plasmodium sporozoites into the human. The sporozoites enter the liver and reproduce asexually and release the merozoites into the bloodstream. The merozoites multiply inside red blood cells and are released, and the cycle repeats and repeats and repeats. Certain merozoites develop into gametocytes, and then from such, the gametocytes are ingested by another previously uninfected mosquito, and the gametocytes develop into gametes and reproduce sexually, forming sporozoites within the mosquito, and the cycle repeats itself. That is malaria by way of Plasmodium, the apicomplexan. So I won't go over the gregarines, but these are the ones that attach themselves in the intestinal epithelium of mollusks, annelids, and arthropods. And then Toxoplasma gondii, So it invades epithelial cells of the human gut. Most individuals infected with this parasite mount an immune response, preventing any permanent damage. However, if that support from your immune system is not there, it could very well cause brain damage. Heart and skeletal tissues too can be damaged. Now we're at the ciliates. So ciliates are unicellular and characterized by their mode of locomotion. So they are heterotrophic protists that use numerous cilia for feeding and propulsion. Each cell has a macronucleus and a micronucleus. The micronuclei are exchanged during conjugation. So they have two types of vacuoles, the food vacuoles for food digestion, and the contractile vacuole that regulates the balance of water. These are ciliates. You're seeing one such example of a ciliate here, and this is a paramecium. A paramecium. Next up, we have SAR rosaria. First up, it mentions the radiolarians, such as radiolaria. They're from the phylum Actinopoda. They have that glassy exoskeleton made of silica and those needle-like pseudopods. Now to the, and I'll go back one more time with the radiolarians. So they're part of what are called the Actinopoda, with that silica exoskeleton. So this is what gives the radiolarians their distinct shape, and the microtubules support the pseudopods that extrude toward the tips of their spiny exoskeleton. Now to the foraminifera. So the foraminifera fossils are what create those huge limestone deposits found across the world, and the foraminifera are heterotrophic marine protists with pore-studded shells primarily formed by deposits of calcium carbonate, which is why they're there for the limestone deposits. So they're rich, such as you find in the White Cliffs of Dover. And there you're seeing the White Cliffs of Dover with all of the calcium carbonate. Pretty amazing to see there. And now the Cercozoa. So like the marine radiolarians, the Cercozoa have silica exoskeletons, but most are found in soils. So the Cercozoans ingest green algae and may provide clues to the evolution of endosymbiosis. Moving on to characteristics of Archaeplastida. So we're here now with those that go through the process of photosynthesis. Rhodophyta. They're photosynthetic, multicellular marine algae. So red algae produce accessory pigments that may give them a red color. They lack centrioles and flagella, and they reproduce using an alternation of generations. And they'll be taught more so in Principles of Biology 2 than being taught now in Principles of Biology 1, that alternation of generations. So this is now showing you the red algae and that beautiful red color. The green algae. So chlorophytes and charophytes are green algae. Unicellular chlorophytes include the cladionomas, which has 2 flagella, and chlorella, which has no flagella and reproduces asexually. So one such example begins here with Volvox, shown to you there. Volvox is an example of a colonial green algae. Some cells are specialized for producing gametes or for asexual reproduction. It may represent a step on the way to multicellularity, and then we have the multicellular chlorophytes can have haplodiplontic life cycles, such as shown here with ulva. Ulva has a sporophyte and gametophyte generation. So those 2 generations, the sporophyte generation and the gametophyte generation. And in this case, they're identical. However, the chlorophytes did not give rise to land plants, although they are the closest relatives to land plants. So they consist of flattened sheets 2 cells thick. So both groups within the charophytes, the corals and the coleocytales, have plasmodesmata, cytoplasmic links between cells. They also undergo mitosis and cytokinesis like terrestrial plants. Are the most closely related to the land plants of all the algae. And if you ask me, they look pretty similar to a land plant. We're at the amoebozoa. Next to last group, excuse me. So most amoebas are free-living, but some are parasitic. Many protists are free-living and found in the soil and freshwater ecosystem, but few species are pathogenic to humans. And you see that pseudopod there at the top right near about 1 o'clock in the figure. Plasmodial slime molds are multinucleate. Plasmodia in their feeding phase are macroscopically visible masses of oozing slime. These colorful large cells undergo repeated rounds of mitosis without cell division. So there you have it, the plasmodial slime molds. Cellular slime molds, they exhibit differentiation, and that's cell differentiation. Cellular slime molds can signal and interact with neighboring cells, resulting in differential cell types in an aggregate organism. And this is showing you that cellular slime mold here. Now to Opisthokonta. Colonial conoflagellates are structurally similar to freshwater sponges, and molecular similarities have been found. Fungi and animals are more closely related to each other than to plants because they share a common ancestor, which leads them to be grouped as the Opisthokonts. And as I mentioned, of particular interest are the Opisthokonts are the conoflagellates, the unicellular organisms that are most like the common ancestor of sponges, and indeed all animals. The conoflagellates have a single emergent flagellum surrounded by a funnel-shaped contractile collar composed of closely placed filaments, a structure that is exactly matched to the sponges, which are animals. These protists feed on bacteria strained out of water by their collar, and the colonial forms resemble freshwater sponges. The close relationship of conoflagellates to animals was further demonstrated by the strong homology between the surface receptor, a tyrosine kinase receptor, found in the conoflagellates and found in sponges. This surface receptor initiates a signaling pathway involving phosphorylation. And this is showing you a colonial conoflagellate resembling their close relatives, the sponges. This has been the Protists. This has been Principles of Biology 1.